Hello and welcome to PhDtalk. My name is Miriam and I'm the host on this podcast where I interview PhD students to let them talk about their topics and struggles and joys, and where me and others share tips with you to make your life as a PhD student more effective, less stressful, and most importantly, more fun. For today's episode, I interviewed Hongzhen. Hongzhen is a PhD student at Ghent University in Belgium, and we know each other through a EU Horizon project that we are both part of called Nutritious Cycle, and that project focuses on finding ways to close nutrient loops in agricultural production systems. Hongzhen's research investigates bio-based fertilizers derived from animal manure, and she conducts experiments to compare how plants absorb the nitrogen from these bio-based fertilizers compared to mineral fertilizers and what it takes to improve their fertilizer value, of the bio-based fertilizers, so they can compete. I hope you enjoy today's episode, that you learn something new, and then let's talk science. Today's introduction is in Mandarin.
大家好，我叫罗红珍，我来自中国，现在在比利时根特大学攻读博士学位，然后现在是处于准备答辩的阶段。然后我在博士期间所做的课题是农业废弃物的资源化处理。具体来说呢，就是采用一些传统和先进的技术，比如说厌氧发酵、固液分离、氨氮吹脱，或者是膜分离，这些先进的技术去从畜禽粪便中回收氮磷钾这些植物养分，然后并把它用作有机肥，施用在农田当中，以评价它的一些经济和环境效益。通过这个方式呢，我们可以实现这个养分在农业系统中的有效循环，然后实现可持续的发展。然后目前我是作为一个研究工作在做一些欧盟的地平线的项目，然后目前是在执行2个 Horizon Europe 项目，所以今后也是希望能够继续在科研工作中去发挥自己的一些特长。
Hi, Hongzhen.
Hello, Miriam.
Thank you for having this interview with me today.
Thank you for inviting me.
In lovely Ghent. Would you like to start introducing yourself a bit? Like, what did you do before you came to Ghent in Belgium? And a bit about your background.
Yeah. Well, I studied my bachelor and master in China Agricultural University, that's in Beijing. So that's 6 years in the same university. And my major was agricultural engineering. It's more or less similar to what I'm doing now. But before, I focused more on the agricultural system and focus on more on the technology to recover nutrients from bio-waste. And now my work focuses more on the application part, like apply the recovered nutrients to the crop and evaluate the efficiency.
Okay.
Yeah.
So now we already went into your PhD that you do at the University of Ghent, and you said that you are looking at recovered nutrients and their application.
Yes.
Could you explain this a little further? What are recovered nutrients and what does application mean?
Yeah. As my focus is mainly on nitrogen. So nitrogen is a very important element in all the life, and also it's a kind of big part of the pollution in the environment. So within agricultural system, we have input to the system and we have output. Like, for example, in a broad view, the input is from the lightning or the precipitation or some biological fixation from nitrogen gas into ammonium nitrogen or nitrate nitrogen. This one can be absorbed by the crop. They use as fertilizer and the crop grows and it feeds to the animals. But animals cannot fully digest it, and some of them come out as manure. And manure can be used as fertilizer, but somehow if it's accumulates a lot, then it's also a pollution. So our task is to make use of the part of the nitrogen inside the animal manure and to use some technologies like anaerobic digestion, release the organic nitrogen from the manure into ammonium nitrogen so that it can be used by the crop. And some other technologies like stripping or scrubbing, membrane filtration to more efficiently recover effective nitrogen as fertilizer. Then this fertilizer will be again applied to the crop so that crop can use it as a nutrient and then grow again. So that is a closed system. Our aim is to achieve the closed loop of the nitrogen inside the agricultural system.
Okay. So you're trying to be like the link between the manure, and the manure is too much, so we cannot efficiently use all the nitrogen because it's polluting the environment instead.
Yeah.
So you're trying to turn the nitrogen into a form that is more like manageable.
Yes.
Okay. And you mentioned some of the technologies like stripping and scrubbing and membrane filtration. Could you tell us a bit about like what that means and what it is?
Yeah. We start with anaerobic digestion. Yeah. That's one of the very common used technology to produce biomethane, biogas containing biomethane and some other carbon dioxide gases. These gases generated from like when you put a manure or other bio-waste into anaerobic conditions that without any oxygen. But there are some kind of bacteria can make use of this organic material to get their energy. And in the meantime, the big molecules will break down into small molecules. In that way, we get biogas. And in the meantime, there are some leftovers like liquid or some minerals inside the liquid that we call digestate. Yeah, that's one of the products. And basically, anaerobic digestion is the primary treatment for the manure. And then, like what we said, solid liquid separation and stripping, scrubbing will come out after that. Solid liquid separation is kind of easy. That you may use some centrifugation or screw belt press to separate the liquid part and the solid part. And stripping and scrubbing, that's based on the principle that we have ammonia and ammonium. So that's NH3 and NH4. They are kind of in a balance in the liquid fraction. If we add some other conditions like increase the temperature or increase the pH, then the balance will be adjusted. Like with higher pH and higher temperature, more nitrogen will convert into ammonia. That's NH3 form. That's a gas form. So if we again use some airflow to blow it out, then this ammonia will come out to the air, and then we can use another solution like acid solution to catch it again, so that to form another pure mineral product. We normally use it to up concentrate the nitrogen from the waste streams.
Okay.
Yeah.
So in the waste stream, the nitrogen is too diluted, like there's too many other things, too much liquid.
Yeah.
So you're trying to get as much nitrogen out of the liquid and then transfer it into another product, which is then very concentrated in nitrogen and therefore easier to handle, right?
Exactly.
Yeah.
And why we use these technologies? Because if we apply the, let's say, manure or digest directly to the field, there are quite some problems. One thing is the transportation. Like you said, the nitrogen concentration inside probably very low, and you have to transport very large quantity, volume to the fields. And that cost a lot of money and also time, and also maybe some carbon footprint during the transportation. And also when you have the solid form, like manure, there are basically during the intensive livestock production, farmers will add additives into the feed for animals. For example, some heavy metals content feed. So by the end, the manure will also contain high concentration of heavy metals or antibiotics. Yeah, we are still in doubt if this would cause higher impact in the environment. So it's better to avoid applying this to the crop.
Yeah, makes sense. Yeah. And then the third technology, the membrane.
Yes.
Nutrition, yeah.
Membrane technology is actually a very broad thing. My math topic was also on the membrane technology. And actually there are a series of membrane. So the first, we can divide it into two kinds. One is mainly depend on the pore size of the membrane, and the other one is mainly the pressure driven membrane. So the pore size, we have like very briefly, the micro filtration is the size. I don't remember. It's like 50 micromilliliter. The pore size larger than micrometer. And we have nano filtration. That's a bit smaller. The pore size is at the scale of nanometers. And for the pressure-driven membranes, we have like a reverse osmosis, or have forward osmosis. So it's depends on where you put the pressure on. Of course, the energy consumption and also the efficiency of these membranes are different. So you have to choose, depend on what's your target and what's your feedstock to the membrane.
Okay. Yeah, I see. Thank you. It was a very clear explanation, I think. So now we are at the point where we have our kind of product, right? We are up concentrating something.
Yeah.
And now it's your turn, right? Now you're trying to figure out how can we bring this back to the field and how will it actually behave in the field, right? In relation to crops.
Yes.
So what is it exactly that you're trying to do?
Yeah. So during my PhD, actually, we do get the products from the industry scale. They have produced the product, and then we use it to evaluate how is the nitrogen released from the products and how the crop can take up it. I had done some lab scale experiments and field experiments. So basically, we applied certain amount of the products into soil, and from three aspects, I would look into the nitrogen efficiency. First is just apply to the soil without any crop effect, and is in the lab scale. Let's see how the microbes can affect the nitrogen efficiency, because like I said before, the nitrogen in the natural environment can be different forms. They have organic nitrogen, like what we know about amino acids. That's one kind of organic nitrogen. And we have also mineral nitrogen, that's ammonia nitrogen or nitrate nitrogen. That's the forms that crop can easily take up. So, but in the products we used, they are combined. Like we also have organic and mineral forms together, but our target is to maximize the mineral forms of nitrogen. So I use the laboratory setup to see just with the soil microbes, the function of soil microbes, how much nitrogen could be converted from the organic into mineral. And when we have this information, and we will add the effect of crop, that's still in the controlled conditions in the lab scale, but we have the crop, because the crop can react with soil and microbes they have a very complicated relationship. It's competition. They are competition for the nitrogen and for the nutrients in the soil, but also they are helping each other because some of these microbes, they may lack of carbon. They need carbon as an energy source, and crop can release some easy taking up carbon from their own body, like through the roots. So in that way, they are also helping each other. We would like to see with different products if the relationship between the crop and the microbes could change, if they could towards more competition or towards more cooperation. So I use some PCR tests to see the functional groups of microbes inside the soil and see
PCR test.
PCR. Yeah. So the microbes, they have cells, and inside we have DNA.
Okay.
So the PCR test is just to multiply the detected DNA of the certain microbes.
Okay.
Yeah. So in that way, we can detect the population or the abundance of these kind of function groups of bacteria.
Okay. You can see which DNA strains are there, and then you can see this DNA strain belongs to this type of microbe, and then
yes,
okay.
Yes,
yeah.
So in that way, we figure out this group of microbes probably increased after applying the products, and the increase maybe help the crop to get more nitrogen. So there are quite some different connections and links, but that's a macro system between microbes and crop. That's a small system. And then when we have an idea of how it's happening in the small system, we scale up to our field system. That's in a field scale. We apply the same fertilizer and see if similar things happened at the field scale.
Okay.
Yeah.
And what did you see? How did the microbes react, or how did the crops react? How did the interrelations change?
Yes. So, when we don't have any crop in the soil, we applied fertilizer. Already from the literature, we know that the mineral nitrogen could be released in very short term, like 1 month already. That's most of the nitrogen, if they are going to release, the microbes can be quickly function. But with crop, this is another story. In my experiments, because we want to see the effect, if our products can replace the commercial fertilizer. So we control the nitrogen application rate to a kind of a lower rate, so that it's not optimize the requirements of the crop, but actually with the commercial products, commercial fertilizer, it can satisfy the crop's needs. But we want to see if our product can also satisfy the crop. So, for example, normally they apply the commercial fertilizer at 200 kilogram per hectare. But we want to see that's kind of in Belgium, actually, it's very high rates, and it causes some residues in the soil. So in our experiments, we reduce it to 150. And we want to see if still with our product, the crop can be satisfied with the nitrogen provided. So, but it turns out in our experiments, the crop actually cannot compete with the microbes. Yeah. So in the lab scale, it seems very optimal for the microbes to generate and to reproduce. So but also probably depends on my crop, because I choose maize, which is a arable farming crop. It's more fit with the broad nature. So in the lab conditions, probably they are not so competitive to the crops in the beginning. And my experiment was not really long. It's like 3 months. So basically for maize, the growth season was up to 5 months, 4 to 5 months in the nature. So yeah, I think that's the reason why in my experiments, the microbes actually absorb more nitrogen than the crop. But overall, our question was answered that our products actually can replace the chemical fertilizer because we didn't see much difference, a significant difference between the treatment with commercial fertilizer and applied with our fertilizer. So they are comparable.
Okay.
Yeah. And again, that's happened in the field experiments. So there was no significant difference. Well, when we talk about field experiment, there are more factors can affect the functions. The weather or the native soil probably very rich. That's the case in Flanders. So we do see some benefits of our products because normally the commercial nitrogen fertilizer is in the solid form. That when you apply it to the soil, it stays in the applied layer of the soil. For example, if you put it in 15 centimeters deep, then it stays. If there is no irrigation or no rainfall, it just stays there. But our product is liquid. So it's more, the mobility is stronger. So when you apply it to it, if there is no rain during the early season, it can actually filter through the soil. And if the roots grows, the nutrients can follow the roots. So that's why in the early season, actually our products give a better performance. But by the end, because during the 5 months of growing season, in the middle, there are some rainfall coming. So the solid fertilizers also dissolved and then reach the crop. So by the end, there was no significant difference.
Okay.
Yeah.
Okay. So the mineral fertilizers could be replaced by these recycled fertilizers, if you want to call them like that. Okay.
Yeah.
And is that done already somewhere, or is that something that farmers are trying to do or interested in trying to do? Or
yes. I would say now we are promoting this kind of let's say bio-based fertilizers to replace the commercial fertilizers. But indeed, we have some problems in the legislation because not all regions or countries accept this kind of products to be applied to the soil. There are, for example, in Flanders, when we recover the nutrients from manure, the current legislation still recognizes this as manure. So it has to follow the same rules as manure. Like in Flanders, the manure can only be applied to the fields at maximum 170 kilogram per hectare per year. That's the maximum. But sometimes for some of the crop, it's not enough for them. If you use commercial fertilizer that's pure mineral, so they can add more. So we don't think that makes sense because you already have 100% and you add more, but we have organic and mineral nitrogen combined in the manure, but we cannot add more. We can only add 170. That's sometimes it's not enough. So and when we recover the bio-based fertilizers from manure, we have again, even though it's kind of a mineral, for example, from stripping and scrubbing, ammonium nitrate, ammonium sulfate, ammonium water, these products from the stripping and scrubbing, they actually are mineral nitrogen. But we cannot apply it over 170.
Okay.
According to current regulation. So we also trying to promote some new rules, some regulations, new rules, saying, we define this as another product, another category that's different from manure.
Okay.
So, in our project, so we have these terms like bio-based fertilizer, that's processed nutrients from manure or from other bio-waste, but it's more efficient than manure nitrogen or manure phosphorus. Yeah.
So ideally there should be a third category. So either a third category, we have the manure, we have the mineral fertilizers, and then we have the bio-based fertilizers. Or the bio-based fertilizers could become like a subcategory of mineral fertilizers, or be even treated the same way, you think?
Yeah, I think we should have differentiate from manure so that we can, because currently what in our experiments, these bio-based fertilizers actually performed more similar to commercial fertilizer rather than manure. So in that sense, it should, in economic sense or economic sense, it should be separated, and maybe it can also help some other regions, like they don't have enough source for fertilizers, and also in these years the price of fertilizer is increasing. So if we can recover these nutrients from manure, in one side we reduce the pollution, and the other sides we reduce the cost for buying fertilizer. So,
and when I hear that, I feel like, okay, that seems quite logical. So do you think that the legislation is just slow, or are they reluctant because of, like, why wouldn't they change the regulations? Is there a reason?
Of course, the things, this bio-based fertilizer, even though we are kind of promoting it, but it's not very well studied. Still, we just studied maybe decades to 20 years or how many. So we need time to accumulate more evidence to prove that this is really efficient and useful. And also for agronomy, normally we have this fear that some effect may come after several years, especially when you say about some accumulation. Nowadays we have heavy metals accumulation and antibiotics and microplastic. These ones you will not see significant effect in few years. So that's also at the regulation aspect. The policymakers, they always want to make sure this product is really safe and efficient. But currently our evidence probably not like enough to support that it's really safe.
Okay.
So we still need to gather more results and do more evaluation to see how this in a long term, and how could we, if it's not, how could we improve? So that needs some time.
Okay, yeah. So that also maybe policymakers can ask questions, right? Like, what else do they need to know? Because now we spoke about nitrogen, right? But obviously there is also phosphorus and many other nutrients, right?
Yeah.
Yeah. Okay, I see. Yeah. And apart from that, what else have you been doing in your PhD?
Yeah, my PhD is mainly for the researching part, is mainly doing some experiments and also cooperate with the projects to kind of disseminate my report results. But apart from study, I also that's as an international student here, I really want to enjoy the life, because I came from, I would say, China and Europe. It's kind of a very different system. So when I first came here, I feel excited to explore the new world and a lot of new things. And in our group, we have a lot of international colleagues. So that's a really good chance to make friends and exchange the culture. I really enjoy that.
Yeah. Okay. Yeah. Nice. It's also something I enjoy about science.
Yeah.
The different people. Yeah.
Yeah.
Yeah. And then did you like experience some struggles to your PhD, if you want to talk about that? Like if the joys were like, I guess the experiments and the colleagues, and was there something that you would have liked to know or that you struggled with a little bit?
Yeah. Yes. During PhD, I think most of PhDs have been going through these things. Like, for example, you read a paper, it's really interesting, but you find, that's what I want to tell. It's already published. So I have to find a new idea. So that's something like struggling. But also when you have some experiments, especially when we are doing crop, that's something you cannot really control. It's life. They have to grow.
Annoying.
So sometimes they grow good. Sometimes they grow bad, but you don't know why. Maybe you cannot find the rhythm. So when the experiment failed, or kind of the data was not so nicely as you expected, then you're like, it's nightmare. Yeah,
sure. I mean, even though we try to control everything, it's still living beings, right? And of course they grow differently.
Yes.
Yeah.
But interestingly, this is also something you get fun from. For them, like I said, when you find a paper, even though it's published your idea, but it's written well and it looks very interesting, you feel satisfied because you get new knowledge. And I really like that when you find you know more about the world, you know more about science. So that's a sense of satisfying. Yeah. And also when you set up your experiment and when you see the crop is growing, wow, that's really like, oh, my baby. Yeah. It's a sense of accomplishment. I like it.
Yeah. Nice. Yeah, sounds good. And then would you like to stay in academia? I feel like you are quite excited about science. So
Yeah,
you'd like to stay.
Yes. Personally, I like the feeling of doing science. Yeah. Even though I also get bored when writing all these reports. But that's the life of the researcher. So you have the positive side and also sometimes negative part. But yeah.
Yeah. And what is it you would like to explore in the future?
I still feel I have quite some to learn about soil. So I want to go further with the soil science, because when we see our world, it's like a broad world. You see different living things, human, birds, trees. But when you look into the soil, it's also a very broad world. That's different microbes. It's just like different species of human or animals. So it's really interesting, I would say. And they are playing a very important part in agriculture. So I want to learn more about them and see how they could help us to produce more food or more efficiently use our resource.
Okay. Yeah. Okay. So also combining them with bio-based fertilizers or just understanding them better so we can foster their abilities kind of to help us produce food. Yeah, makes sense. It's a nice vision.
Thank you.
Yeah. If someone would like to get in contact with you or learn more about your research, how could one find you? I
have a ResearchGate page or a LinkedIn page. Then so I can share with you. Maybe you can just find it, or you can just search on ResearchGate my name, full name over there. Yeah.
Okay, perfect. Thank you. Is there anything else that you would like to share with us? Any thought?
I would say, for PhDs, what I found really important is doing sports and traveling. That's one of my hobby. Maybe it's a hobby. So I think like now in our group we have every week swimming or badminton or some soccer group. So after work we go for sports and then release some pressure from the work. I think that's a good way to get your mind away a little bit away from the work. And then also during my spare time, I like to travel around with myself or with other friends. So because I really love the nature, so I went to when I was looking at the computer, I feel like my life is just inside the screen. But when I go to travel, I feel this is more broad life, and the life is so beautiful. I think that's kind of make you think more positively over the difficulties you met during the work or life.
Yeah, I see. Do you think it's particularly difficult to come from like very far away to Europe? Do you think that makes it a bit more difficult, or?
I would say the COVID really make it more difficult. Yes. So before that, it's even though it's far, but now the transportation is so convenient that you can anyway, if you feel homesick, you just find a flight home. But during COVID, there is some different issues that I didn't go home within 3.5 years. So that was a little bit tough for me. But now also the technologies, like video calls, you can get in touch with your family, with your friends.
Okay, so it was okay for you in the end, kind of. Apart from COVID, but if other people like follow now, then they wouldn't hopefully experience something like that again.
I feel we are, even though with COVID, but we are more lucky than people like 50 or 30 years ago who don't have any source to get connected.
Yeah, definitely. Yeah, sure. Yeah. Thank you for sharing your thoughts with us. That was really nice.
Thank you.
Thank you very much. Good. Bye-bye.
Bye.
Welcome back. I hope you found today's episode insightful, interesting, and that you learned something new about bio-based fertilizers and the different processing technologies there is to improve their values, their competitiveness. And if you enjoyed this conversation, then feel free to share it with your friends, leave a rating, drop a question or a comment in the discussion section. And we are always happy to engage with you. And then I'm looking forward to the next episode. And then I wish you all the best. Please take care of yourself and if you can, someone else. And then, bye-bye.